the global network. I'll say that again. Welcome to Frugal Science. Let's wait for a few more minutes. I'm seeing lots of names across the screens, people that we have known for a long while, people that are new to this class, and I'll describe the logistics a little bit. We will dive into what I wanted to cover today in the context of oceanography. And then for the folks that are in the international cohort, if you can stay on a little bit after the class, we'll do a little bit of an introduction, and I'll have everybody introduce themselves in the next class. So let's just give people maybe 1 or 2 more minutes. While we're waiting, maybe let's use our chat. If people can write the country that they're connecting from, that might be a fun thing. I want to figure out how many of you are sitting in the middle of your night. You're actually really sleepy. I'm looking at Webho right now. Uh, and you're still here. So if you can just type in the chat countries that you're connecting from, it'll give me a sense. And you can be more specific if you want to be in terms of locations. I see Tunisia. I see Maria from Uganda. Let's give people more time. So while we wait, I'm just going to wait 2 more minutes before we start officially. Some quick sets of logistics. This is a global class. Everybody participates, but this is a class really. Oh, this is cool. Kevin has a country poll, so if people can fill out the country poll. It's a participatory class. So one of the key things and an idea is you can still listen to all the lectures. You can still connect through the content. But for you to really take the class, the idea would be is that you will be involved in a project heavily. There is a local cohort on Stanford campus, and then there is an international cohort, but the teams are built across no matter where people are. So you will all be building teams across the globe. We will choose specific problems, although we'll talk about lots of general problems, but a successful project is something where you'll pick up a very specific problem to tackle and solve. So I think I just want to give people a sense of the participatory side of this. And after this class, I'll also just answer questions around for the students that have joined for the first time. Usually, I often do a big introductory lecture, but I'm going to save a little bit of time because I'll mention why in a second, and I'll do this introductory bit in a little bit. Any other logistics? Yes. So for the next till April 20, the class will be on Zoom. This is really for the Stanford cohort. After that, we'll meet in the UTL, the teaching lab. But of course, all the international participants can still participate via Zoom itself. And one thing that we will do is next get together, we will have people just do line of intros. And actually, maybe I'm wondering, there is a Notion page online, and I think, Kevin, if you can find the Notion page and share that link, everybody should write a brief introduction from a team's point of view. Otherwise, it'll take too long a time. They should fill out a brief introduction for, I mean, we are happy to also just open up your answers for what you wrote of why you wanted to take the class. But it'll also be useful on Notion for people to fill that out. So
I think I might send out a survey instead at first, and then we can share out the results on Discord. Yeah.
yeah, something I think I was thinking about for for team building and getting people to be used to the Notion platform. It's
Okay,
useful for everybody to create a short profile, essentially. That's what I really meant.
thanks. Okay.
Uh, okay, that's all for the logistics. There's a lot in the agenda, so I just want to get started. Um, I am curious, it's amongst the folks. I think just quick Zoom logistics. Um, if you can, please turn your videos on. It's kind of fun to be able to see people that are listening, people that are participating. It's perfectly okay if you're pipetting and running an assay or doing science in the background as well. I don't mind. But it is useful to see who's there. And then the second part of this is also you all are familiar with Zoom, so we'll kind of manage the audio side of the story. It's perfectly okay to interrupt and ask questions. Just raise your hand at any point of time. Uh, but at the same time, just if you're not speaking, make sure you are muted. Okay, I think we'll begin now. The first thing I want to do is we had covered in the last kind of a lecture a little bit of the logistics of how the class runs. We will cover that again very briefly, but we will really focus on oceanography, challenges in the ocean, and thinking a lot more about aquatic water bodies for this week. And I want to talk a little bit about sets of solutions and criterias around sets of solutions today. So we'll focus much more on the solution side of the story. So this will act as a case study for what we are trying to do. But it would do disservice to many of you who are connecting for the first time uh, where I'm connecting from. So I'm just going to give you a little preview of what's outside my window. And it's a great introduction to why we're talking about the oceans today. Um, at some point of time when we go out, I'll show you a little bit of operationally what happens.
I think by virtue of being on the ocean, the connectivity might be a little off, so we'll wait a moment.
Everybody is frozen on my end, Lee. And okay. Um, could you all hear me for the last couple of seconds or no?
I think maybe if you repeat the last 10 seconds. Yeah.
Okay, I just said I am not technically stuck, but I am stuck on a boat for the next week. And it's a perfect place to talk about oceans and ocean challenges per se. Um, so I think let's start with a thread around, I'm just going to share my screen. Uh, we covered most of the logistics. Um, But overall, in terms of thinking about spaces and questions we will be covering as modules for the class, um, I think there is a lot of global health challenges that many of you are excited about. So we'll definitely have a module dedicated to that. I'm really hoping some of you will pick up projects in the context of health and especially health for all. Um, we will definitely cover certain sets of threads around environmental monitoring. This has kind of been one of the key part of what's been called One Health or planetary health. Uh, Treyansh, your mic is on. Can you please mute yourself? Kevin, can you do that?
Yeah,
Uh,
I muted them.
okay, all right. The other bit on this front is we'll cover a lot of science education threads, and one of the things to think a little bit about is if the class of problems or ideas that you're excited about are not in these bins, that's perfectly okay. Spend some time with us, talk to me specifically, and we'll just expand these. I think these are certain starting points, primarily because there's certain sets of time and resources we can invest, and it's fun when many people are working in close by scientific sets of areas. But if you have sets of ideas that you're excited about for this class that fall outside this view, it'll actually be useful to talk to me and some of the TAs beforehand itself as well. So we're focusing really on the oceans. And last class, which we'll post online, we had talked a lot more about kind of oceans by the numbers, what the scale of challenges in the oceans feels like. And one important thing that we will try to do is talk about the solution side of the story a little bit today. A couple of logistics. I think overall, in terms of projects, it will be team projects, 3 to 4 people. We use Notion as a way to track projects. So we'll send out a message in terms of how we document projects. But it's very important to use Notion as your open lab notebook. I think the more you share with each other, teach things to each other, the better you'll realize actually that there are tremendous number of skills that are present in this room. uh, some of you I have known for a very long time, and it's really remarkable sometimes that when these skills actually intersect and meet. So use Notion when you're writing your description. Do share and write certain kinds of things that you have access to, certain types of capabilities that you're trained on. So really, we'll use Notion as a team building page itself as well. And I think slowly you'll all get to know each other too. Um, Discord is really for much more of the chit chat, trying to get ideas on the table, quick conversations. Every project will have Discord access itself. Uh, but an important page very quickly that I want everybody to start thinking about is this thing called the idea board that we put on Notion. And the idea board is where you write and share things that you are excited about. We are primarily here to help, but a successful project is really driven by students across. And so it's important if some of you have already filled out, uh, we carry over idea boards from last year, the last couple of years. So you'll see that document itself, and it's very self-explanatory. It's perfectly okay. Oh, go ahead. Was there a question? Okay, no, I'll continue. Um, okay, that's all I want to say about logistics. One last thing, I think it was mentioned in my email, but I just want to reiterate before we dive in. And I know I had mentioned this to the Stanford cohort itself. Uh, we are a very and culture here rather than a but culture. And an and culture is people will share all kinds of ideas with each other. You will share vulnerabilities, you will share challenges that you think about. So it's very important that we understand that and we build upon other people's ideas. You know, it's very easy to kind of turn down ideas. It's very hard to add upon them. And so kind of keep that in mind every time you're giving feedback to each other. It's very important to kind of focus on the and culture. Okay, that's all I want to say about logistics. Any urgent thread? Otherwise, I'm going to dive into what I want to cover today. And I'm eager to get to know all of you, but I want to make sure that we do that post the class. Okay, I'm not seeing any hands, so let's talk a little bit about what we want to cover today. Um, So, I think I want to start with something much more personal, which I mentioned that I am in the middle of the Pacific Ocean right now. And I had not anticipated this or thought about this, that we happen to have chosen a station. We call a station essentially a location that we are studying, right on the edge of the Great Pacific Garbage Patch. And it's just so ironic. I mean, we planned all of the sets of things. We planned location, we planned logistics of what we wanted to study. We're primarily studying deep sea life. And every time I started doing the toes, so now these are toes that I'll talk a little bit about in the context of instruments. An instrument that we use to do the toes is called Mockness, and we deploy it 1 kilometer deep. And it's been 5 days now, and we have deployed it 4 times. Every single day, a significant portion of our catch is plastic. And I'm not talking about surface toes. I'm used to surface toes. I'm used to seeing plastic in the surface toes. So this is in the middle of the ocean. And of course, for some of you who might have heard of the garbage patch in the news, it's not as if you see garbage floating in the ocean, although we have seen that too. We've seen buckets floating here. Uh, we are around 500 miles from the San Francisco coast. Uh, but what has been very surprising on this expedition specifically is how much plastic we are seeing in the deeper parts of the ocean. And I think this is something that I would love for a few folks to pick up. uh, it makes the challenge of plastic removal, I would say, infinitely harder. Because, I mean, I think we have known for a while that it's a volumetric problem, but I just want to share this because I'm really surprised by how much plastic. So now we are going to try to quantify this to really understand volumetrically how much plastic we're seeing. But every little toe, and I think it's important to understand that at the end of the day, there are certain sets of ecosystems that once we have essentially entrained, and it's an important question to also think about scientifically why there is so much plastic in the deeper parts of the ocean itself. Because technically, we had discussed this last time, density for most plastic is actually lighter than seawater. But because there is some amount of biomass that grows on it, it actually becomes denser. So there is a lot more in terms of thinking about it's not just plastic per se. There are microbes and other sets of organisms that are growing on these sets of plastics. Uh, and I think uh, this has been documented from time to time. And kind of this was a New York Times article, a couple of years ago, thinking a little bit about what it's called. It's snowing microplastic. And I think viscerally seeing that for the first time here it's quite dramatic. And I think I'll send some and upload some pictures from the expedition and also the pictures of the plastic that we are pulling out. We happen to have a Raman spectrometer on board, so we are trying to classify now which sets of plastics are we collecting. But an important thing to sort of remember is sometimes the physical experience of a problem is really what motivates you to work on something. And I know there are a few team members joining from Mumbai, and literally a month ago, we all had an experience where we were in the Thane area, which is the largest natural flamingo gathering in the world. And there again, we saw some of the largest plastic pollution that I have ever actually seen. And so, uh, if somebody wants to, you can share on the chat a link to kind of the nature reserve that I'm talking about. So, what I'm trying to say is problems, and especially problems in the aquatic ecosystems, are fairly easy to notice once you start paying attention. And I think there are hundreds of examples of these right in your neighborhood. And since we are going to be focusing on aquatic technologies, it's valuable for first you to map what type of aquatic ecosystems are around you. So folks that are on Stanford, Lake Lagunita might be a place to start, or Jasper Ridge has a pristine lake, which was formed because of a dam. And interestingly, that has lead in it in the bottom. So if you do a soil core, you will find lead and you will find massive deposits of lead. So sometimes ecosystems that look so pristine on the surface are still deeply polluted or perturbed. And I think as an exercise, it would be important for all of you to find, life depends on water, find an ecosystem locally and just, have a little look to get a sense for what might be the state of that ecosystem. And we'll talk a lot about mapping the state of these sets of ecosystems. This is a photograph Adam from the lab just sent me. It's on KQED from last night. Um, some of you are in the Bay Area, and minke whales are washing up. 8 whales have washed up in the last 2 days or 3 days. I don't know many of you have seen it or not. Uh, Nobody really knows the cause for something like this, but a couple of years ago, we had what was called an algae apocalypse in the Bay Area because the Bay Area has this incredible massive estuary, and there was a explosion of algae that was toxic that killed hundreds of thousands of fish across the bay. And I think it's just another reminder in some sense of most ecosystems, even when they look pristine on the surface, might be being highly impacted and above a critical point where you will then start seeing larger scale issues. So it would be very important as an exercise for this week for all of you to identify one local aquatic ecosystem. I know some of you are connecting from IIT Bombay. You have your own Lake Powai. I would love to get a sense for you to go and check and see uh, what can you decipher about your local ecosystems. Uh, the big thing we will focus on today is really the framework on what lives in the ecosystem and what makes some of this ecosystem actually work. And I'm assuming, actually, can people do a show of hands? You can do a thumbs up. How many of you have done microscopy regularly? Just uh, just a thumbs up. So I'm seeing lots of hands and not everybody. Um, okay, I think maybe 50% of you have done microscopy. And so almost all of you have experienced this fact that if you take a little drop of water, you find this incredible life forms. Um, the ocean is a little bit like that. And I think one of the factors that's been at play currently in understanding some of these ecosystems is the ecology in these sets of ecosystems is far more complex. We don't really have good frameworks for understanding that ecology quantitatively as yet. And one of the big threads on our front has been thinking a lot more about how do you quantitatively map aquatic ecosystems with their biodiversity and see whether you can correlate that back to many of the sets of problems that we were talking about. So this type of an approach is often used in the context of using biology as an indicator. And so the the picture that you're looking at is, drawn as a mandala, but it's essentially a consortium of more than a million plus species that would live in our water bodies. And effectively, in some sense, they are both indicators. And at that same time, these are the sets of things that process most of the either the nutrients or the sets of pollutants that you would actually put in a water body itself. Uh, And the big challenge for us had been for the longest while is we don't even have a map associated with who is where. And so kind of the project that I'll focus on today and talk a lot more about is really a tool to be able to map some of these ecosystems in an automated manner. And so first of all, why would you care about mapping what's in our oceans, for example, at microscopic scale? Or in your lakes, for example, at a microscopic scale? There are a couple of answers to something like that. Um, One of them is literally associated with this plot on the right. I think I've mentioned this plot on the right a couple of times, but it's worth looking through. Uh, what you're looking at on the right is, the x-axis is time, 1850 to 2000. The y-axis is the CO2 flux. And that red curve on the top, can you all see my cursor? No? You see my cursor, right? Okay. Uh, you see, this is the rise of CO2 as a function of time. But what's interesting is the one at the bottom. Everybody has known this. We know this very well because we know how many barrels of oil we sell every year. So that's the fossil fuel emissions. The bottom curve is actually really interesting because the bottom curve gives you what we remove out of that every year. And there are lots of estimates, but the one we will focus on is the ocean sink. And you can see 20% to 30% of this CO2 that we have emitted has actually been removed by the ocean. Interestingly, the estimates are that as we put more CO2 in the atmosphere, more of it is absorbed by the ocean and more of it is somehow removed. And we don't really understand this process of removal, and we study that process quite a lot. This is called oceanic carbon sequestration. The same numbers also apply for land and other mechanisms. And then you can see the light blue curve is really what is the remaining carbon, and which is what we call as much of the CO2 that accumulates in the atmosphere. Now, the process of this removal is really driven by plankton. And this is really why we care about trying to understand who's there, what does the biomass look like, and how is it distributed along the depth of the ocean. Uh, Similarly, another example to take for this would be the notion of the kind of models that we currently build. And, these are some recent papers. One of the challenge has been is we have not been able to balance our carbon budget, especially for the ocean. It's very hard to balance carbon budget on other places, but if you think about it, if it's one of the most important thing that we care about, our budget should be balanced in the sense that you should know every molecule of carbon and where it goes. And that still remains an open question, which is a challenge. So for many a times, sets of places, and actually there are many other budgets, atmospheric gas budgets that have never been balanced yet. And I love the honest title of this top paper, which this is a published paper. It just says what the dash is wrong with our present carbon budget calculations. So if any of you who are going to think about sequestration, especially either ocean or other sets of sequestrations, it's really worthwhile reading some of these papers. Because ironically, much of the IPCC reports have no biological parameters. And that's concerning because if you think about it, all the oxygen that we breathe, half of that comes from cyanobacteria and much of in algae from the ocean. You can imagine life perturbs the planet so dramatically, but we have very little control in terms of thinking about how to account for it in our models per se. So that was sort of the starting point for us in thinking about uh, this framework of frugal oceanography is, can we bring measurement tools for the ocean and aquatic environments to a broader group of people? That's how we started this framework of thinking about mapping planktonic ecosystems. And I'll primarily focus on two tools today. One is OpenCTD and the other is a Planktoscope, which are both open source initiatives now. One of them we started, OpenCTD was started by another team. Both of them are really good examples of how a single project can then lead to a large amount of data first collected by communities in order to address and understand the types of changes that I was talking about. So I think we're going to dive in and I want to show you the sets of instruments and what the instruments look like, um, and tell you a little bit of the history of how we got started on building this tool. We were spending a lot of time in the ocean, and I used to always use this instrument called flow cam. And flow cam is essentially an instrument that allows you to do something like this. And it always felt very rarely would the boats have flow cams. It would cost roughly around $100,000 to begin with. And that was our starting point to ask ourselves a question, could we actually do something better, but at a significantly lower cost? I have Mohammed's hand up. Mohammed, do you want to ask a question?
Yeah, hello, Mr. Romania. Hello, everyone. Sorry for interrupting, but just
No,
I
and
have,
Mohammed, before you should introduce yourself and then ask a
yeah,
question.
all right. Hello, everyone. My name is Mohammed Al Snefer. I am a freshman student from Tunisia, which is located in North Africa. I'm happy to join this course. As we all know, frugal science is an important uh, science when it comes, I think, to create, let's say, a solution, affordable solution to everyone. So I'm very happy to join this course. So,
Thanks, Mohammed. Can
uh,
you ask your question? Yes.
yeah. So as I saw in the previous slide, plankton can produce 40% of the world's oxygen. So can we make this, let's say, bacteria with an artificial way produce it? Can we produce this?
So I think you got cut off there. One of the threads to think a little bit about is the thing you're hinting towards is, can we control some amount of both the biomass in these aquatic ecosystems, and can we use that to actually control our planet's atmosphere? So, I think the the short answer is lots of people have been thinking about it and talking about it, and there is a great experiment that has happened since the 1970s. It's called the iron fertilization experiment. The results of it are mixed. So the idea came when, if you look at the map of Africa and you look at the Sahara Desert, what you notice is there is a red color across the ocean next to Senegal, Ghana, all across. And that's iron dust that's literally floating from Sahara and landing in the ocean. And earlier oceanographers had noted that always the satellite blooms that happen occur around there. So that led to an idea that, wait a second, it's possible that if iron is one of the most common nutrients that gets depleted very quickly when a bloom happens in the ocean. And that led to an idea thinking about, could we artificially do this? So you're right, Mohammed. And we will talk a little bit about this in the context of when we talk about carbon sequestration, uh, that people have done experiments, and especially the experiments are happening around in Antarctica and regions where some of the most productive oceans are. Uh, it's a scary prospect. It's pretty tricky to think about that we are going to completely modify some of these ecosystems. And the reason it's scary is we don't have any measurements. So most of the time, even satellites that are looked through, satellite images don't really tell you the depth of what happened. And so often enough, it doesn't tell you explicitly. And this is one of the key problems. I'm really glad you asked this question. When you have a million species interacting with each other,
Yeah.
do we feel comfortable? Do we have the kind of scientific knowledge to perturb or engineer anything? Imagine engineering a solution where you are perturbing one thing, but you have million other parameters that are not in your control in a highly dynamic system. You would never call that engineering in any way, and you would never proceed with it. But unfortunately, we have arrived at this point that even some of the most conservative oceanographers that I know are currently considering these approaches, primarily because we are left with no choices. So I think, I mean, it's
Yeah.
much more of a arriving at this realizing, and I think our approach has really been is while other people think of these sets of approaches for perturbations, what can we do on measurement? Could we really build systems that are very specifically, in a very precise manner, actually measuring these ecosystems at the largest scale? So at the least, we would know what is changing in these microbial ecosystems, and simultaneously, we would have monitoring in place when we have to become more aggressive about perturbation of our planet. Uh, I mean, it's also important to note that uh, we are perturbing our planet. So there was a phenomenal paper that was published with COVID. Many of you heard about, I mean, the air quality in Delhi was significantly better, if some of you know that. But interestingly, the shipping vessels that stopped, they emit sulfur dioxide. And that is reflective. And actually, you could see it that some of that pollution helps mitigate heating in the ocean. And so, ironically, I mean, it's going backwards now that the emissions from those ships was suppressing uh, because the ocean absorbs 90% of all the heat that we generate. So it's not, none of these knobs are one way. And so I think the big idea to think a lot more about is the fact that we have to understand the complexity and the dynamics of these sets of systems. I don't think we are anywhere close to be even thinking about perturbing anything, but at that same time, it feels urgent to monitor. And I think this is why I said you should all find one aquatic ecosystem around your home or where you live right now. You'll get a much better sense of how hard this is, how dynamic it is, because otherwise it seems like, oh, we took a data point. You come back tomorrow, everything has changed. So it would actually be really valuable from a perspective of, and you can store samples, and we'll talk a little bit about sets of tools. Even if you create an archival record of, say, a season, every week, a sample collected from a lake fixed, that by itself is actually very important in being able to then look at a pattern or a trend. Okay, so I'm going to continue, but and again, feel free to interrupt. Uh, depending on how much time I have, I'll take all the questions or I'll take questions in the very end. But you should always raise your hand, type in chat. Um, okay, so I think since I've already mentioned a lot of things about this, I'll just tell you a little bit of a history of how this project got started, more to give you a flavor of, sometimes it does take 5, 6 years to get something done. But at that same time, when you take this community-driven approach, some really interesting things happen. So, uh, several of us in oceanography got together, starting to discuss this notion of, initially, one of the goals was that our oceans are so large, and the vessel that I am on currently, for example, is very expensive. So expensive in the sense that it's 60 to $70,000 per day to operate a mission like this. So 15 days, you can multiply that by 50 or $60,000 per day. That's a lot of money. And we asked ourselves a question, could we engage sailors? And, there's statistics that's out there. There's roughly 15 to 20,000 sailors in the ocean. And this was a project first initiated by Colomban and his group in France, where they gave a very simple thing, essentially a plankton net, a little bit of a filter, and a pump. And this was initially really designed around genomics. And this was, I think, 11 sailboats. And you can see most of them originated from one town in France. That's right here. This is Brittany. Brittany is known and famous for sailing. So no wonder and no surprise that Brittany was there. There's lots of places in New Zealand. But what was fascinating is you can literally see the tracks now. And these are individual families, by the way. These are not professional scientists. These are individual people like you and I, maybe a little bit more adventurous, maybe, that go out around the world and they collected sets of data. And this was primarily a genomic database. And we showed at that time in that paper that encouraging and engaging citizens in an approach like this can lead to mapping of microbial data sets. Now, one of the problems at that time I had noticed was genomic sequences don't really evoke the kind of emotions and don't really give the kind of cellular state that's needed. And so at that time, I started this project of building a kind of an automated low-cost system that allows us for do flow-through microscopy. So many of you have used the full scope. This is an example of now an automated system that will have continuous imaging. The one on the left is I'll talk about that as an original instrument, but that led to this project that we call Planktoscope. And I'll show you the very first prototype that we built in the lab. At this point, it's a large project, and I'll get back to this slide in a second. But actually, let's start with this instrument. So this was version, I would say version 1 or version 0. You can see, I'm going to play this for a second. This is on a sailboat in Gorex, and you can see an imaging system on the side. You can see a basic pump, and there is a filter cube in there. We are operating it on a boat right now. It's built of, Thor lab parts, something that we could put together very quickly in the lab. Now, this took us, less than 15 days to put together, and we were able to take it on a boat to test something out. This was when Hong Chuan was in my lab and Thibaut had just joined the lab, and we built this version 1. Now, one of the first things that we did is we gave this version 1 when it was barely working. And I think, again, I'm using this as a case study, so it's important to understand when you're thinking about projects to realize sets of decisions that you make. It's incredibly important for projects like these to go into hands of other people that will be the actual users as soon as possible. And there happened to be a sailor named Gurek who was passing by San Francisco, and he's I would say, a mad person, mad in a really wonderful way. You can see his trajectory of his sailing. He's the youngest person to cross the Cross Canadian Passage in the winter time. So, you see this dotted line. This is the most treacherous sailing route because it's completely frozen. And I'll show you some of his footage. But I had asked him, and then Thibaut wrote to him, and then he ended up stopping here. We spent some time in the lab, and we installed the very first planktoscope in his boat. And I'm going to show you just the footage that he sent me uh, to give you a sense of what this individual sailor was trying to do. So this is drone footage. He's all by himself. And he carries a chicken. I think it's more for company, but also it's a good protein source too. The eggs, you don't eat the chicken. If you eat the chicken, you're done. But I think it's a reminder sometimes that this is just one person who's sailing across the planet. And the idea is that what if each one of these individuals can actually do the kind of measurements that we need in our ecosystems? It's also a reminder that we should spend more time outdoors. So on this boat, we got the first data sets for what it meant to install. So that is his boat. We've installed the machine. He's doing the plankton tows. Uh, the boat capsized a couple of times. He survived, he was okay, but it was quite rough. But we learned quite a lot from that as an experience to think about what do we need to do for the instruments to survive kind of these types of oceanic conditions. That then led to version 2 of the instrument. And here you can see the instrument became a lot more complex because we were trying an idea in modularity where different people could build different modules. So now you can see it's become this what is that candy called? Trombone, the triangular chocolate. Uh, and then just like
Toblerones.
break those pieces. But this is now operating on a car battery. Uh, Thibaut and I took this to Lake Chilika in India because Lake Chilika is one of these pristine ecosystems with incredible bird sanctuaries. This is now uh, another scientist in Lake Chilika who's been studying this ecosystem for a while. And one of the things around this expedition was it taught us that the instruments have to be far more robust. But it was also a good reminder of thinking a little bit about what type of community we want to be using this tool. So that was, I would say, version 2. And then finally, eventually it became version 3, which is what is on the site now. And I think another kind of a big lesson as a case study is we spent more time documenting this tool on the website than writing academic papers about it. So if you go to the Planktoscope website, you can find every little detail. I mean, at the end of the day, this is just a Raspberry Pi with a camera module and connected to a pump, two little motors for focus. It's a fairly simple construction. There's lots of space for other modules. There is a GPS module that people have added. The entire thing is completely open source and it's operated with Node-RED, so very easy for other people to add different sets of modules on. Initially, we were using these EBD slides. Some of you who work in the lab, they're super expensive. Then we replaced them with glass capillaries. And then kind of, this is what I meant by the instructions. And I know some of you have planktoscopes at this point and have used them. I'll ask you guys to do a demo at some point of time, especially WebHub. It would be fun for you to run a demo for the class because you happen to have the unit that you've just constructed. Uh, so these are important lessons to remember in terms of building a project because it has to work in the hands of not just us. So when we build scientific projects, we're often thinking about ourselves, we collect the data, but this is the type of data that this machine now collects. And so one of the important aspects is that you can get, so this happens to be a dinoflagellate bloom. You can see just one specific species passing by. You know, an important aspect of this is that there is a tool chain around quantification of all of this data. And one thing that I'll show you is before I show you the data, now this is the current unit. And I think, you know, some of you have already seen it. This is a unit that's assembled by Fairscope. Thibaut, when he finished from the lab, we started an entity in France now for manufacturing this. It's still completely open source. All the parts are online. Anybody can build it. But he also makes a kit. And what I love is one choice that we made as a design choice. So just to give an analogy, uh, this is the little glass capillary. You can all see that. Uh, the capillary costs between, you know, I think 30 to 40 cents. What's interesting is that it's a square capillary. It's not a round capillary, so you can actually image through it. Uh, there are little magnets everywhere. It'll couple through. Here is the little lens that you can see. It's an M12 lens you can find on Thor Labs or Amazon. There is a Raspberry Pi 4. There is a peristaltic pump, and that's it. And then there are two of these linear motors that you can get on Taobao. Um, the There was kind of a joke in the lab around let's build it out of the same material as boats are made out of. So this material is actually really interesting. I'll pull out to show you. Uh, this is called bamboo plywood. So you can see it's all bamboo, but it's plywood. But what's really neat about what we were able to find is it's machinable. different types of wood is not machinable, but this is machinable to an extent that, you can see I can make some very precise uh, parts. So usually I would use Delrin or aluminum or something when I want to build high precision parts. These are so high precision that I can even make sort of mechanisms out of it. So this is, I mean, we tried many different materials. We tried a material for cutting boards. Initially, all the units were made out of plastic, but at the end of the day, this is what stuck. It was a fantastic choice because this material is now being used for sustainable flooring. So lots of homes have bamboo flooring, and this is just the flooring material. Uh, and again, in that framework of torturous path, it's not that we solved all these problems at the same time. We actually solved them accidentally from time to time. Um, I'm going to now share some data to give you a sense of what does it take to convert a project into how do you handle the pushback? So one of the first things that people said is like, oh, this is great, but can you really collect quantitative scientific data? Uh, because, it's a fair point. Can these citizen scientists who don't have any training in science in the past, could we train them? Could the instruments help and support? So that started a journey with Fabien and Villefranche and a couple of us starting to convert these videos into very, very quantitative data set in an automated pipeline. So the pipeline now has been, shown, but the pipeline is pretty straightforward. What you're looking at is this is the raw data that it collected. This is color correction, simple threshold filter. That's an object classifier. Wherever there are pixels that are lit, we break them out. We upload all of this segmentation data to another platform called EcoTaxa. EcoTaxa is really designed around a classification system. A big ML engine has already been written. So you can feed all of these objects and out comes species names, which is really exciting because then you can do something very quantitative. So these are the two machines that we compared. This is Planktoscope on the left. This is FlowCam on the right. And I think it's very important, just as another lesson for projects, is we can't be shy about, we really have to compare ourselves to the best that's out there. So some of the instruments that we'll be building and designing in the class, the goal is to be able to compete in the best of class. So in this object, for example, you cannot tell the difference of which photographs are taken from Planktoscope and which is taken from FlowCam because they are explicitly equivalent. And that's an important goal to have because people have this perception often that, oh, because it's low cost, it must not be good enough. But what you're trying to do is you're trying to make a design choice to ask yourself a question of uh, what is the quality that you actually need for the scientific question that you're interested in. So that's an important thing to take on very seriously. Uh, Again, this has been quantified now. It's online. I like this plot. It just sort of shows you uh, all of the organisms that it found in, say, first 5 minutes. And on the right corner right there, if you look very closely, I think it's hard to see. I'm going to zoom in on this. Uh, it's kind of fun to look at visualizations, but zoom, zoom, zoom. Can you all see? Can you recognize what that is? Anybody wants to shout out?
Plastic.
Actually, we could play a game, Where is Waldo? And see if you can find. Anybody wants to shout out, just raise your hand, who can tell me what is this object what organism is this?
Is it a plastic?
Exactly. It's an organism of our making. That's a microplastic. When you see sharp edges, when you see these jagged edges, they're very rare in nature. Um, Many of these are dinoflagellates. You can see there is a diatom, another dinoflagellates. Uh, Now, this is really interesting. I mean, and this is low resolution on my screen. but the pictures are much higher resolution. You can see a pinnate diatom attached to marine snow. You see hundreds of species of copepods, another dinoflagellates. You see right there diatoms. Uh, Many ciliates are here. And one of the things is you start seeing these broken shells. So these are dead diatoms. That's another diatom. There's a Serratium right here. All of these are now diatoms. That's another Serratium species. There's a tintinid right there and another microplastic. So now you have a better sense. Do you see that shape? Classic sign of microplastic. Um, and so one of the things is that, now you can immerse in the data and you could quantitatively say something about what you're observing. And that's an important aspect of trying to transition as you're building certain sets of tools to really focus on pinning down so that when two people make measurements, they can be comparable. And that's not trivial. I think I would say we've spent half the time in designing the instrument, but half the time and majority of the most recent work, especially with Fabian's group, has really been on mapping and making sure that the tools are calibrated, auto calibration protocols. So I think sometimes in the design process, you might realize that you spend a lot more time refining the tool than inventing it. And you have to have wins in your sail to continue doing that because sometimes it can be very arduous as a process and you might feel I'm not making progress, but indeed you're making the most important progress. So another big lesson to keep that in mind, that a successful project will continue for many, many years to come. So now these are the new units. That's Thibaut right there. Now he was in the lab for a couple of years and now he set up a manufacturing facility in France to make many planktoscopes. This is the new unit. These are the data sets that come out of the units. Again, microplastic right there. And maybe in a couple of weeks, I'll kind of do a quiz. If some of you have spent time in your ecosystem, we'll try to see how many of you can identify many of these. It's important because you also need to build an intuition. And again, this is the key aspect. I could have given you names of these from genomics, but you don't have the visceral understanding of what that ecosystem might look like. So this is another ecosystem, and Adam in the lab is kind of, we have these little postcards that we send people, but they are from data collected by communities and often done in a manner that they are geostamped. So this is another, many of you can recognize Pyrogyra. But there is a lot of quantitative work that's gone in comparing. This is the last transect that Fabian's group did, going from Punta Arenas to Laurent and being able to map one-to-one between our data sets and FlowCam data sets to show that the total biomass that we are measuring is exactly equivalent. This is an important characteristic because we care about quantitatively how much biomass was in the water column. And I think one other aspect of this and again, we will have lots of discussions about open source in the class. I'll have Ethan in the lab give a lecture on, when you all sign up your open source licenses, there is a lot of legal language. It's important to understand it. So I'll have Ethan give a short talk about what that language means, for what project, how should you choose thinking about what to make open. Uh, the design of the Planktoscope is open. And so when we posted the website, people started sending us these pictures. This one was made by a UCSF professor. Another one was a carpenter who just wanted to build this. Many people started using wood by themselves. And, this community has been growing. And if any of you, wherever you are interested, this is a good, I would say 10, 15-day kind of a project to build and assemble. I think I'll have the Stanford cohort assemble some of the units, and then we'll see if there is enough interest, and especially if a project gets chosen that's utilizing Planktoscope for a new capability, we'll think a little bit about seeding some communities. I know Ashoka has one Planktoscope community. IIT Bombay has another community. But this is what the community looks like now. And one of the nice thing about this is nobody's in charge. People do what they want with the unit. That's important as a project because sometimes you might seed something, but it's important for people to take them in different directions. That's an important lesson in a project itself. And again, it doesn't have to be an explicit goal, but for something like this, if we care about a large number of people engaging and mapping the ecosystems that they live in, it's important for them to have agency, and that agency is important. This is the quantitative data that's online. Again, we don't put incentives on people to upload their data. We just request them that it would be nice if all the data is collected. So we've contributed a lot of data. Many other people contribute data sets, and these data sets are growing at this point. And kind of as a summary, what's been really interesting is there is roughly around 300 or so units now, 25 countries, 26 million images. And one of the things, it still is in the academic community. So I would like the price to be much lower. The next challenge for us is, it still costs around $800 in parts. I would like this to be around $100. And so any of you who is interested in kind of building, I've been calling it Planktoscope Mini, which would be a miniature version of something like this, lower compute power, but much of the architecture remaining the same and simplifying some of the design, a purely manual focus mechanism, for example. Uh, and then I think one of the things that's happened is that there is a much broader community, also just publicly, that shares data. On the architecture side, what's been really interesting is Ethan in the lab has been writing a lot of architecture code to connect planktoscopes with each other. So now there is an online repository where you can go, and so if Daniel is sitting anywhere he is, can control the planktoscope that I have in another community. And it's kind of a fun way of people kind of, I think some of you know about the project where computer time and clock cycles are given to other people. And similarly, people can log in into another person's machine and essentially operate it. It also just broadens the access. But the big thing that we are trying to do is make this network much more accessible. The other big project in the lab is to make this instrument in situ. So one of the things so far is that we still collect samples. But if we had the capacity, we should be able to do something like this completely underwater. And I'll show you, this is a collaboration with CCA, with Adam in the lab, where we are now trying to make small buoys. Uh, and these buoys have the planktoscope with a tube hanging at the bottom. This has a solar panel, has a battery. And again, this is project in progress, but one of the key ideas is to be able to make this completely autonomous. And then this brings me to kind of another platform that's worth just mentioning briefly. Uh, while you will be designing and building a module for a project, other people will be doing other things, and sometimes there is a lot of synergy. So we were worrying quite a lot about power and data, but in the same time, there's a company in San Francisco so far that developed these plastic buoys that already have data connectivity and solar panels on them, and they are ready to then tether any instrument to. So what's been valuable is, for a project that we thought will take many years for us to finish, now has all the modules done, and all we have to do is connect to their modules. And this module is called Bristlemouth. So for many people who'll be using oceanography as an example for low-cost oceanography projects, we have a collaboration with SOFAR. I'm going to try to get a few buoys to test, but it would be fantastic to use that platform, because then we don't have to solve power and communications as a problem. A very useful platform, and I think we've been using it for all kinds of things. Okay, so I think that's all I want to say about Planktoscope. Maybe let me say one word about OpenCTD, and then we'll open up for questions. Um, yeah, okay, let me say two more words about Planktoscope, and then I'll jump. So going back to the big thing that I was asking before in understanding our ecosystems, one of the insights is that uh, microorganisms are incredible bioindicators. And this has been shown for a long time. And in the old days, before we had chemical sensors, we used to use bioindicators as an approach to understanding something. So, when you have high bloom of Cylindritheca, it's associated with, say, very, very high nutrients in the water column. When you have uh, Ketoceros, you can associate that with certain sets of frameworks. And it's valuable to kind of think about and map ecological conditions mapped to the biological indicators. There are a couple of ways of doing this. One of them is satellites. And so now there is new data that's coming out with hyperspectral satellites, which is all accessible. Satellite data has become really cheap. But what's missing is most often you don't actually know what the attributes were on the ground. So when we say chlorophyll A is not a chlorophyll B doesn't mean you have to actually attribute that data to what was physically happening on the microscopic scale. So one of the big things is these correlated data sets will become very important because you want to be able to tell what does the satellite see physically. Uh, the other side of this story is also driven by other measurements, and that brings me to this tool that I'll just briefly spend time on. I really like the community. This is a tool that I have not been associated with, but I admire how the project has been run, which is called OpenCTD. One of the things that we also have to do is make measurements that are environmentally anchored to the species that are there. OpenCTD is a platform for anybody to build for roughly around, I think, $800 or so to build. This has state-of-the-art conductivity, temperature, and depth measurements. So if you were sitting on top of the Mariana Trench, you would want to know conductivity we use for salinity because you want to know how the water and the salinity of the water is. What do the temperature profiles look like and how deep they are? So how you're correlated. So every oceanography expedition has expensive CTDs. They cost $30,000, $40,000. So this is a really fun project. It's very well documented if you take a look at it. The sets of sensors. I love this photograph because it sort of sets the stage for anybody just sitting out there making sets of measurements. Uh, and again, this also gives an indication for the kind of instruments that you can put on fishing poles. Much of the documentation on OpenCTD is online, so I'll let you all read that. But I would say it's a fantastic framework for also when physical parameters are to be measured. It's a good platform because it already has lithium batteries incorporated, has all the chassis for data recording. And so if you're thinking about one specific type of measurement, you can essentially add to the same platform itself. Okay, the community is documented. Let me end with this and then let's open to questions. I think the lessons in terms of thinking about instrumentation and specifically instrumentation in kind of the ocean perspective is there are certain sets of modules that you're trying to design. You won't be able to design all of them. So you want to rely on an ecosystem. We wouldn't have been able to build a Planktoscope if somebody else had not released a Raspberry Pi. Just not possible. Similarly, other people can't build other things unless we had released this as a tool. So there is modular aspect of it. There is applications, which is we don't know when you invent tools what the most powerful applications will be. So it's valuable to have a vague idea, put instruments out there, and let other people build on the application layer. So right now, these tools are used in oyster farming, abalone farming, They're used for identifying algal blooms. We have a vague idea early on, but you're not going to be able to pin down completely. And then the last bit of this is really uh, releasing projects such that they can have a global footprint and have a community that supports each other rather than one single team trying to manage that is critical, because at the end of the day, the projects primarily survive because many, many more people are interested in taking that idea forward. So that was sort of what I wanted to share. There is a lot more we can talk about in that space. Uh, let's turn this into a dialogue rather than a monologue, which I don't really like. Uh, Questions, comments. This is sort of the open mic. We do this every class. End of every class, last 10, 15 minutes are open mic. People can ask questions, comments. You can share sets of ideas you might have around aquatic ecosystem and aquatic ecosystem monitoring, for example, or challenges in the context of the ocean. You can ask me anything about the discussion that we had. And then after that, we'll transition to do quick introduction amongst the international and the global cohort itself. But this is open mic, so feel free to ask or initiate a conversation.
I just had a quick question about hi.
So Kamal, you have to introduce yourself, say what you do, where
Yeah.
you're connecting from, and then comment or question.
Sure. I'm Kamal. I am a PhD student in my final year. I'm from TIFR Mumbai, which is right by the Arabian Sea. So another ecosystem that I will look forward to exploring very soon. But I just wanted to ask you, like you said, you're I don't know how many miles away from the Pacific Ocean, which is
Yes.
near the San Francisco coast. So, how do you figure out these permissions? Because, as far as I know, for the Arabian Sea, we are not allowed to like go and collect samples. I remember Obaid used to do that, and we have heard stories about people going from TIFR collecting samples down the seashore, and that was allowed. But right now we have security. So, how do you manage that logistics?
Yeah, I think there are marine protected areas. You have to abide by local rules and laws. Um, Marine protected areas have separate sets of permits. If you are really setting up a scientific study, it's actually fairly straightforward to get permits. Like, for example, Stanford campus. You might not realize that, oh, for collecting samples on Stanford campus, there is a permit. But it took me less than 15 minutes to write the email, and now I have permits for the entire campus. And it was not that there is a permit, it just so happened that there is a salamander species that is a rare species that is only on campus. And the only thing that they wanted to do is let people know that if you accidentally pick up salamander eggs, just put them back. That's it. So often enough, it's fairly straightforward and simple. And there is a positive. And I think, this is an important point that you're raising, because Nagoya protocols and many other sets of things are coming in place. There are certain countries, Indonesia, New Zealand, very difficult to work. I mean, even any microbiome, very difficult to actually sample and publish. So an important thing is to just anchor and look at what the local rules are. I mean, fishing licenses are given all the time. For most non-marine protected areas, sampling is actually fairly straightforward. There are certain areas that might have other sensitive threads, and I think this is a cultural issue itself as well. Air pollution used to be like that, when only government regulated bodies could release the data for say black soot and particulate matter. But is that the right policy? This is also something we have to ask. And so if you can only hear from the official source and not really a distributed monitoring systems, how do we believe? I mean, this is an issue around journalism. So it's an important thing to think about when that pollution and when the insights are important as measurement tools become more and more widespread. How do we as citizens organize around? And it's fairly straightforward to then think about a citizen science permit. Imagine a process that you're not applying it as a professional scientist. You don't have all the backing around it, but you are applying a permission as a citizen scientist. Now, as a country, any set of a country, would a country actually support that? And, to a certain extent, it's an important policy discussion to be had as well. As individuals, you should all kind of, check, but, you have to be stewards of those local ecosystems. And in fact, then you are in partnership with the entities that are trying to control them.
Makes sense. Yes,
Raghav, go ahead.
I'll surely check with people in TFR and get back to you with that.
Yes, I think it would be fantastic, and it'll also help many other folks who want to do coastal sampling, especially
Absolutely.
for microplastic. I mean,
Sorry, I cut someone. Someone was trying to say something.
No, go ahead. But I think, Kamal, this would be a homework on your plate because you'll help many of the other participants from
Sure.
India. Okay, Raghav, go ahead.
Hi everyone, I'm Ragat. I'm at Stanford and I'm a senior in bioengineering. So I had two questions. The first of which is, you've been mentioning how there's very large differences in the ecosystems in the ocean, but I was wondering if we have like a quantitative measurement of how, like, how much spatial variation there is. So what the optimal sampling frequency would be. Because you're talking about how we would not want to sample, we'd want to do continuous reading. What are the like time or spatial points we would want to take those measurements at? And second question is you were telling us to like engage with the ecosystems around us. Is there a way to get access to the fold scopes, perhaps, so we can do that?
Yeah, I think two things. I'll answer the easy one first. You all can have as many full scopes as you want. That's trivial. I think once we know the stable cohort and who are the sets of people that are here for the whole class, we'll take addresses from everyone and ship full scope kits. And the folks that are on campus, it's pretty straightforward. And I think one important other aspect of this would be is the spatial temporal. That's a very hard question. We don't even know that answer even for a lake, for example, or because you have to think about temporal dynamics of these ecosystems. They have natural dynamics, and unless you do very high frequency sampling, only then can you figure out what the right sampling frequency should be. The ocean as a spatial is so large, and our data sets around the biological indicators are so limited that we don't even have a good sense of being able to do that calculation. For example, Argo floats is a good example. There is roughly 4,000 to 5,000 Argo floats. I think I talked about Argo float in the second lecture. Some of you were there, and the others I'll post all the lectures online. So you guys should listen to 2 of the intro lectures. Argo floats is interesting because I think people say this that, oh, 4,000, 5,000, it's the highest resolution temperature record of our ocean as a function of depth. But still we are 100 to 1,000x because the heterogeneity in these ecosystems we only discover when we do much more fine grain measurements. Otherwise, these heterogeneities are not even known. So if the heterogeneity is not known, it becomes very difficult to be able to estimate some of that. It's a good exercise in terms of thinking about measuring an ecosystem at a very high frequency to then be able to say, I think we have got the right spatial and temporal frequency. The problem with the ocean is that the water body moves. So in some sense, you are in the frame of reference of the water body. That's called the Lagrangian rather than the Eulerian mapping. And you have to be in the Lagrangian frame of reference for that water body to have any predictable time series. So if you have your classic bacterial growth assay, it is in log phase, but that water body is the Now, if you do the same thing, I want to watch what does the log phase of microbes look like in the ocean. The physical water body, because there is no vessel, is actually moving. So if you remain stationary in a geographic location, you cannot say something about the temporal dynamics. You have to be in the frame of reference of the water. And that's why Argo floats float. And this is why we're building a Planktoscope that's going to float. It's not that we will still do the Eulerian is going to be people, but Lagrangian will be floating instruments. So, and I think it's when we do the by the numbers, Ragad, it would be really fun if you take that as a puzzle and see for any, say, a large lake, what is the approach to this problem. But as far as I know, I think this is quite a hairy problem. Okay, Ashish, go ahead.
Hi, can you hear me?
Yes.
All right. Yeah, thanks for that introducing us to the ocean world and very inspiring talk. I had two quick questions. One is an easy one.
And then
You
Ashish,
said,
can you introduce yourself and some
oh,
context?
sorry. Yeah, sorry. I'm Ashish Priya. I am from University of Cincinnati. I'm an engineer and teacher myself here, and we're very interested to see other people in this community. So yeah, I had two quick questions. One was easy one, which is you said that you want to make the Planktoscope cheaper, or the next version to be like a mini version to be cheaper, less than $100. I'm curious to learn about what the most expensive parts are right now on that device and why it's $800. And more, the harder question I think is, how do you get money to do these cool stuff, right? Like a lot of the like the build of material might be cheap, but the R&D that goes into and time spent in iterating to a place where it works requires money, right? Like I was curious what your take is to be successful in that.
Okay, I see your point. Easy question and a hard question. Easy is pretty simple. You know, the primary cost is really when you look at the bill of materials, it's just a Raspberry Pi. Everything else in terms of parts is fairly straightforward. It's just lots and lots of little parts. And I think once we optimize that, with Foldscope, there is volume. With Planktoscope, as yet, we have not reached volume, right? It's only still 100, 200 units in a couple of years. That's too small to get any volume. When you think about cell phones, the reason they are so, at least some of the cell phones are accessible is primarily because they have the volume. I think in some sense, there is still a lot of things to be optimized and no project is ever finished. So we're still redesigning Foldscope all the time. We're still redesigning. I think it's just a little bit about how long are you willing to stay with an idea? How long can you keep pursuing it is actually important. Because sometimes we think it's done and then we put a full stop on it, but it needs constant iteration. On the funding side, I think that's a pretty much harder as a challenge. On one hand, everybody is talking about these large scale problems, but we want to jump towards sets of solutions while we don't even have the framework to understand these larger scale problems per se. So sort of our role, all of us in the community, is to convince many of the funders that this approach to science and monitoring and measurement and problem solving is actually scalable, and eventually that's what they look for. I think the challenge has often been is everybody wants to support innovation, nobody wants to support scale.
Yeah.
And there's a classic language of valley of death in the middle, because once you reach some amount of scale, it's also self-sustainable. So at this point, people can also buy a fully assembled kit, and that supports the salary of all the 3-4 full-time people that actually work on the project. So you can achieve some amount of sustainability with scale too. So it's an art, it takes time, and yeah, there's no one button answer, but often enough, if you build fun things, people do come. And so there is some amount of support. I think one thing we have initiated on experiment.com, and some of you have actually been funded through that, is small amount of funds to start and initiate projects. For projects in the class, I'm going to try finding some sets of funds, so dedicated funds associated with that. More and more funding agencies are starting to explore the micro grant model. But microgrants, the problem there also is that it starts an idea on a whiteboard. It doesn't let you do it because, I mean, everything that you saw is 5, 6 years of our time, 3 or 4 people continuously working, and that is a significant chunk of money, right? I mean, just salaries itself alone is a significant chunk of money. So I think maybe we need to be more convincing, which we are not right now. Okay, Mohammed has a question. And then I think I will let other folks go after this question. And if people need to leave, by the way, it's perfectly okay. And after that, if some of the international folks can just stay briefly, I'm just going to do quick logistics.
Yeah. So hello everyone. I have, let's say, a couple of questions. So the first one, how to make a solution like Planktoscope as cheap as possible. So what is the source of these types of inspirations? And it is all about when it comes to choose the device materials or in designing, in the designing parts.
Yeah, I think, I mean, the short answer is you have to be embedded in the problem that you care about. There's plenty inspiration all around us. The real question that, and this is a very important, it might sound trivial, but I'll say this. The real thing is, do you have the time? Unless you can dedicate time to a problem, it doesn't get solved. So we're all inspired. We have all kinds of ideas. There's obviously so many things that need to be done. Does it bother you enough to a point where you can say that I am going to dedicate that much time to the specific problem? So your investment, frankly, is your time. Often enough, we have in our peripheral vision lots of problems that we are thinking about, and you don't focus on them primarily because you're already tied up with many other things that you're trying to do. So you have to let go of something to do something else. So I wouldn't say we don't have a inspiration problem. There's plenty of ideas floating around, like just the internet has exploded sharing of ideas. But often enough, we somehow hesitate in diving in and committing time to a specific problem. That's why I started the class by just making you feel bothered by something. Something has to bother you enough for it to matter.
Yes.
That part is hard.
Thanks a lot.
And you should only work on things that bother you. Don't work on stuff that, other people will handle that.
All right.
Okay,
Thanks a lot
Kamal,
for
go ahead.
clarifying. Yeah.
Yeah, I just had a quick question. I wanted to ask in response to Ragad, I think this thought came to my mind when she was speaking about how you could actually correlate so you mentioned using the organisms that are there inside the ocean as a biosensor for assessing some of the parameters. And I was thinking about it, they are also continuously evolving, right? So you
Yes.
mentioned that in response to nutritive conditions, some of them are enriched several folds. And so that will, their capability of responding to nutritive conditions will also evolve over time. So how do you factor that in when you use them as a proxy? Because then there's a debate whether this is actually changing or that.
Yeah. This is a fantastic question. It's very complicated. So first of all, just some evidence that yes, things are evolving. One thing that's very interesting is they are restructuring and evolving as organisms, but they're also finding new niches to begin with. Like, for example, for plankton, it's known as the planet is heating up, higher and higher latitudes, we're starting to find tropical organisms. microbes that were only found in closer to the equator and the belt are now found much higher. Water is much more, there is much more nutrients there, and at that same time, it's warm enough for them to survive. We don't really have a good map, and I think I think this is a project that's ongoing in the lab. We have done these two pieces separately. We have done genomic mapping and we have done image-based mapping. And what we are trying to do is to do that together. So not only do we say what we found, we actually have enough correlates to be able to say genomically or just even transcriptomes of what do they look like. That's different. So currently there is a project in the lab where we are trying to trap the cells that we image for later access, but to do a one-to-one correlated imaging sequencing platform. Now it's going to take time and it's more complex and it adds more resources to it, but it's an important question because and again, I think this is something that the Foldscope community is also doing right now. When I talk about Foldscope, it'll become obvious for some of you who have not used it. We've now released this card system that people actually save the sample that they image so that at a later time, if it turns out that you found a new organism, not as a species, but at a new place, which was not there reported before, you could actually sequence it. So it creates this archival fossil record of microbes that citizens collect. And this is sort of where the trading cards come in. And so several of these types of ideas to make sure that we have some archival record because it's a combination of low tech and high tech, and sequencing is incredible, but it's harder to distribute it. So we can centralize it to a certain extent. So every sample that was collected, say in Mumbai, can go to TIFR, somewhere close to Bangalore can go to NCBS. And so there can be lots of local hubs for making sure that we are also collecting sequencing data. And I think, unfortunately, many of the initiatives that got started in microbial mapping took kind of only a centralized approach. They don't have a distributed approach. They don't have communities engaged in it. So I think it's a choice that we are trying to make where these two approaches are not orthogonal. They're actually quite complementary to each other. Okay, I'm seeing it's 12. Unless there was any other burning question, I just want 2 minutes of people's time who are on the. Everybody else is also welcome to stay. But one thread that I am noticing is there's lots of people on the call, but I don't have their videos on. So if folks that are joining from outside, otherwise people that have to leave, that's not an issue. I know it's very late already. We'll connect, but I just wanted to make sure that from a timing point of view, and what we will end up doing is in the next class, if people can fill in. So now I'm just turning back to logistics again. If people can fill in the Notion page and please write down a quick intro, because the whole reason to take this class is to build teams that work together. And that only happens when people get to know each other. So we're going to do some icebreakers amongst the larger group. I wasn't sure how many people would actually connect. And I know some of you, this time might not work out. This is like some late in the morning right now for many of you, and it's perfectly okay. But I think if for people who are excited and are anchored and want to stay uh, and want to take, it would actually be very useful. And there is still some time. If you feel that you are all alone, like, I don't know, curious Carlos, where are you connecting from?
Hi, Manu, from Mexico.
Yes. So, for example, if you're the only person from Mexico and you thought that, oh, I have 2 or 3 students or friends around that I might, it's perfectly okay to recruit some local hubs as teams too. But it's also okay to be an individual connected from anywhere too. But what's very important is we'll do some icebreakers soon to make sure that people get to know each other. But the best way to do that would be is if people can document on Notion. especially write down your interests. And in the next class, we're going to have everybody do quick line of intros. And so make sure that you sort of know in advance, because it can take an entire class to just cover the intros, but it's also something personal to at least hear from all of you. We will also cover intros by when you ask questions. So it's very valuable but let's do that with Notion first, and then in the next class we'll kind of start doing, because within 2 weeks' time, I really want us to anchor into teams and some area that you are interested in. So it might feel really fast, but I'm telling you, the quarter ends very quickly. So it's important to start gelling and forming subgroups and sub-teams. So we'll take next class for that. We can also do a separate Friday session, which is really about project discussions. Because at the end of the day, I can tell you all kinds of stories, but it's only useful if we anchor and we actually take on new problems to work on, and especially you all take on new problems to work on. So that's all I wanted to say. I'm hoping many of you will be able to connect. If you cannot do a project, it's perfectly okay to listen to lectures, join in from time to time. But folks that end up staying, it really is, this is a, what is it called? It's a contact sports. So this is a contact sport class. It's not a observational class. You don't watch it from sidelines. If you want to watch it from sidelines, it's perfectly okay, but we have videos for that. Uh, but if you're joining in and connecting every time, I am expecting that you will engage deeply in a project and you will have the time and bandwidth. Because the problem becomes sometimes that if you've committed to a team, they are relying on you. Like Vaibhav might join a team that is based on, say, a farmer's issue in Haryana, and then his team is relying on Vaibhav's kind of participation. Sometimes the other way around, you might be the only person with skills in embedded programming, and then your team sort of starts to rely on you. So it's very important that if you join a team, that you are committed to it. Because if you are only peripherally engaged, then it's a disservice to that team. And there's we will still be friends. I know all of you have lots of on your plate. This is literally your third or fourth job that many of you are doing. So there is it's just only for the teams. And I think we will still have kind of open houses. We'll still have open sets of discussions. But I just want to emphasize that it's because when I see, say, 30, 40 participants, I'm really hoping that, oh, we can build 10 projects. And, between 3 to 4 or maybe 5 people per team. And sometimes then if members are missing from the team, then that team gets disadvantaged in some way. Okay, that's all I want to say. I maybe next class I will connect a little bit earlier if people want to spend some time walking around the boat. I know for the Stanford folks, I had given them the tour, but I know some of you might want to see the boat a little bit. So I will, but I didn't want to take the class time for it. So I will connect 10 minutes early next class, which is on Wednesday, 10:30 a.m. So if people connect around 10:20 or so, you get a 10-minute boat tour. And with that, I'm going to say bye, and I will see you next week. And I should be back on campus by Monday, I'm hoping, if everything goes well. Bye, everyone.
Thank you.